Hello, today I'm going to show you how to use a REBASE restriction enzyme database site to create restriction enzyme digest and insert our sequence of interest inside plasmids to use as expression vectors, or to see how it is transcribed and how the protein is expressed inside mammalian system as well, like in mice, or in vertebrate species like zebrafish, or in Drosophila. So today I'm going to show you about GAL4 VPC GAL4 US system, which can be used, and we are going to find out restriction enzyme sites in the GAL4 US system. What is the GAL4 system? First, let's find out what GAL4 US system stands for. GAL4 US, and in Wikipedia, if you see, it's a biochemical method used to study gene expression and function in organisms such as fruit fly. And GAL4 gene encodes for yeast transcription activated protein GAL4 and US upstream activation sequence. A short sequence of the promoter region to which GAL4 specifically binds to activate gene transcription. Let's go to NCBI site and type for GAL4 US. And we are searching in the database of nucleotide. So it gives us the Saccharomyces DNA binding transcription factor required for activation of GAL genes in response to galactose. So we need the FASTA format here to paste in the REBASE site. So we are going to take the FASTA format, select it, and copy it. Let's now go to the NEB website. So we are going to go to REBASE, and this is the REBASE home. So this is the restriction enzyme database. It has a lot of useful information. If somebody works in the molecular biology lab, they need certain sequence data or restriction enzyme which would help them in transcription, or various other kind of experiments can be performed. And this site is very essential to find out what restriction digests can be generated. The most important tool that we are going to use is REBASE tool. I'm clicking that, and I'm going to go to the New England Biolabs NEB cutter to see whether the DNA where we want to insert our DNA of interest has unique sites for restriction enzymes. So we are going to click this, and let us paste the FASTA format. We can choose file from our laptop, or we can use GenBank number, or use the FASTA format. I'm assuming that my DNA is linear. You can, if you're using a plasmid, you can use circular. The open reading frame has been kept as it is, as default, and I'm going to give it a name of bioinfo. And standard sequences are viral plasmid vectors, which are commonly used, and these are listed here. we can select any one of them. Or viral vectors have certain enzymes which are used for transcription, and they're used for forward and reverse transcription, and also for design of primers. So the most commonly used are T3, SP6, T7. We don't need them, so we are just going to submit this sequence and find out the restriction sites. Here we go. So these are one base cutters, which means these sequences are unique in the DNA, in the GAL4US DNA that we have used. If we want, we can use two base cutters, which means these enzymes enzymes are used twice, which we can see that by the red dots that they show. So if you want to insert a DNA of interest, and we don't want a restriction enzyme digest in the middle of the DNA of interest, we'll see whether these sites are unique or not. If not, we are not going to use that enzyme. So we found out whether there are one base cutters or two base cutters, and we are going to treat the DNA that we want to insert with the same kind of enzymes, and so that they generate unique restriction sites and they get inserted and form complementary bases to get inserted. So this is how we generate the restriction enzyme sites and see whether we can use it or not. There are other things that we can find out. There's a help menu which can be accessed anytime we want to find out information. This is pretty much all about it. There is so if we click on any of these enzymes, we find the restriction cut site, and the arrowheads point to how the overhangs are generated, whether they are blunt ends or sticky ends, that is complementary pairs. There is another term called isoscissomers. Isoscissomers is two restriction enzymes which recognize the same site and cut the same site, but they may generate sticky ends which are different in length. So that is the concept of isoscissomers. These are important because if we want to digest a certain piece of DNA with two enzymes, we might consider this, and these are the reaction conditions that are to be used in the reactions, and we can buy these buffers and reaction mixtures from New England Biolab sites. So these are pretty much the conditions that are used. So this gives us more or less how we can use a REBASE site to know which inserts can be generated with the restriction enzymes, and we can tag green fluorescent proteins or red fluorescent proteins with our gene of interest and see where they are expressed, whether they are expressed at developmental stages. A lot of fun things can be done with restriction digestion, and hope you learned something today.